A gente vai começar então. I'm going to switch to English because we have an international speaker today. Andrew Geall has been very generous in giving us lots of lectures. This is his 5th lecture in 3 days. So he thought he would just have fun coming to Brazil. I hope he doesn't get scared. But Andrew Geall is co-founder and chief development officer of Replicate Bioscience. He has over 20 years of experience in developing drugs delivery system as a pioneer in mRNA vaccines and nucleic acid delivery. He currently serves as chief development officer at Replicate, which he's co-founded. Prior to Replicate, he was chief scientific officer at Precision Nanosystems. He also served as vice president of formulations, analytics, and chemistry at Avidity Bioscience and RNA vaccine platform leader at Novartis. He was at Novartis for over 7 years. He holds a pharmacy degree from the University of Bath in United Kingdom and completed pre-registration training in aseptic dispensing unit at St. Helens Knowsley Trust Hospital. He has a PhD at the University of Bath too in non-viral gene delivery. He's also a rower, and he almost went to the Olympics. And if that happened, maybe the history of RNA vaccines would be quite different, because he played a pivotal role in developing all these delivery systems that we use today, that Moderna used, that Pfizer used, and now there is a judicial litigation on these patents that because companies made billions during the pandemic, and he is in the center of 5 patents that is being litigated. So definitely the story of the COVID would be different if his rowing career launched for good. And he still roar every day, and he's a coach of a rowing girls rowing team, and also he's a scuba dive passionate. So you will see some photos, at least the last photo. Yeah, the last one is a turtle that he took himself. So Andrew, thank you so much for all this day. We have been a lot of fun with you here, learned a lot. So thank you very much.
Thank you, Elton. And this is day 3, presentation number 5. I've had a great time. It's been awesome. I love coming in, teaching the students, trying to inspire them to hand the baton over. It's your turn. So this is, to me, this is my second favorite type of presentation. The first one is my career journey with the students, and we did that one yesterday, and that was a lot of fun. But this is my 16-year story with DNA and RNA, but trying to reinvent the gene vaccine and overcome some monumental hurdles. And this is the story of how we achieved it. I've worked with probably close to 1,000 colleagues now on this project, certainly hundreds. And I'm one individual. I get to tell the story, but as I give the seminar, I have memories of all of these people I work with. They all work super hard. It's not just me. It's a lot of work from a lot of people. So please appreciate the work that they put into. So this is the journey. And through the slides and through data, you'll see the journey. There's a few pictures of faces too. So it begins in 2008. This is when I took over, started the RNA vaccine program at Novartis, and we'll spend a few slides getting on how did that happen and why did it happen. And then the journey begins. I measure performance of a vaccine. That's everything. Does it have efficacy, neutralizing antibodies? Is it tolerated? What's its safety profile? How many doses do you need? But the main question for me is it competitive? Does it work the same or better than a conventional technology? Because if it doesn't, we're not going to displace anything. And so the journey's always been about getting up this innovation curve and being able to perform or outperform conventional technology. Going fast is great in a pandemic, but that's all it counts for in the end in the real world when we're in non-pandemic situations. So you've got to compete with conventional technology. So, you know, I'll take you through the journey at Novartis. We got through the efficacy threshold in the animal models. I'll show you some of that data. Some of the work is very interesting and pioneering. You know, we weren't the first to go into clinical trials with an RNA. That was Moderna. They went in 2015 with linear-based modified RNA in an LNP in an influenza trial in a pandemic setting, a single antigen. Data was okay. It wasn't going to compete with conventional technology, but it was the first real clinical trial showing success. Then we had COVID. Not going to spend much time here. The early successes with linear RNA, but then there was a self-amplifying RNA from Arcturus that took a lot longer to come through, but it's showing better durability than the linear systems. And then I'm going to take you through some pivotal data that we just released from my company, Replicate Bioscience in San Diego. My passion is much longer RNA, self-replicating systems. So my journey really began in 2002. It was my first job off my postdoc. I educated in England and I came over on a postdoc. I actually worked on malaria, learned to be a parasitologist, looking at transport pathways of small molecules in erythrocytes infected with malaria. And I managed to get a job at a company in San Diego called Geall, the DNA vaccine company. Phil Felgner was one of the founders. We delivered DNA with lipoplex delivery systems. And I took, in a period of 4 years, 4 clinical strategies forward through phase 1 and phase 2, scaling up the material in the clean rooms, also on the bench as a research scientist, so doing prototypes and then getting it in the clean room, making phase 1 material, releasing it, scaling it further. And those delivery systems were very, very complex, very difficult to scale. 2006, I decided to move the family to Boston, and I started working in pharmaceutical profiling group, and my job was to design the delivery strategies for short interfering RNA. In 2006, Fire and Mello received the Nobel Prize for Medicine for the discovery of RNA interference, and so they were able to show that these duplexes of short RNA, if they're coded correctly, you can get them into cell, they'll bind in a RISC complex, and this will ablate a specific sequence of messenger RNA. So it's a knockdown technology. We were, at that time, focused on liver delivery and rare diseases within the liver. The lead delivery system was this lipid nanoparticle, and RNAi was going to change the world. I was on board. I started my job, and it really wasn't ready. It wasn't till 2018 that this LNP was finally ready to transition. It was a product treating people with rare liver diseases. What was wrong? The chemistry on the siRNA wasn't there. There was a lot of off-target knockdown effects. The potency wasn't there. The stability wasn't there. The lipid nanoparticles were highly toxic in the animal models, and we were screening rapidly to find more and more potent LNP delivery systems to get the dose down lower so that we could thread the needle of therapeutic index, get below the toxicity, and see efficacy. I liked the lipid nanoparticles because for the first time, they were a scalable liposome technology. For the first time, we had an at-scale way of mixing. The these particles, processing them at scale. And so I was focused internally at Novartis on building novel lipids, filing patents, scaling the process. And in 2008, somebody approached me from the vaccine division. This is Christian Mandel. Chiron was acquired by Novartis Vaccines in 2006, and in 2008, they relocated the division to Cambridge and Boston. He is an academic from the University of Vienna, and he published this paper in PNAS where they'd taken a replicon, so a self-amplifying RNA from tick-borne encephalitis, TBE, encoded an antigen in it. They coated it onto gold particles, and using gene gun, they'd gone into a mouse. They vaccinated this mouse 10 or 12 times and shown they could seroconvert the mouse to the antigen using this novel technology. Really just a proof of concept. It wasn't something that they could develop beyond that first experiment. And gene gun is never used by anyone since those times, really. But Christian was passionate about RNA, and he knew delivery was the problem, and he wanted a delivery person to run the program. And he'd heard that I'd worked at Vical, was a delivery person, had a history in the DNA vaccines, and he wanted to try and encourage me to come and work on the program. The first thing I said to him was, I've read your papers, this is interesting, but there's no way this is going to work. And he said, no, we need a scalable delivery technology. And I talked to him about lipid nanoparticles. I told him I needed about a month to go and review the literature on how to make RNA. And so for a month, I buried myself in the historical literature from about the 1980s back to the 1970s, where the biochemists had to scale enzymatic transcription reactions to make RNA so they could bind them with proteins and do crystal structures. From that reading, it was obvious to me that we could scale the technology, that RNA could be made enzymatically. Now, whether we could do it and have stability, there were a lot of unknown questions, but there was enough there for me to think that this was not that risky a move to switch divisions. Now, he offered me 2 postdocs and a couple of technicians, and I was leaving a group of 16 PhD students, half of them in Switzerland, half of them based in Boston, on this pipe dream. My peers thought I was utterly crazy to be moving, but I just felt that there was momentum and there was a great opportunity. I love to innovate, and I couldn't resist. And my wife just said, "You'll be happy, go." And so 2008, I left on this dream and vision of reinventing the gene vaccine. Jeff Ulmer was also there. He was the global head of external research, and we wrote together, I think, 25 papers describing the technology in various peer-reviewed journals and reviews. Jeff was the editor of those, I was the writer, and then the scientists who contributed dropped in as the middle authors. So these are old memories of a great time. So 2008 I began, and this is now my career history to date, and we're going to kind of pinch point on all of these things. The data, preclinical data from Novartis for 7 years. Then Novartis was sold off their vaccine division to GSK, and I left. I came back to San Diego, where I had started and where my wife is from. I did something completely different, delivering siRNA, but this time as a conjugate on an antibody. I've got one slide describing it. There's a whole lecture and seminar on that technology too. It's been highly successful. But while I was doing that, I was consulting with the RNA vaccine field because of my unique experiences, and was able to overcome some challenges by working with this company in Vancouver, Precision Nanosystems, particularly about building out a lipid library and scaling a mixing technology. I went full-time in the first year of COVID there, raised a large sum of money from the Canadian government to build out the platform for a COVID vaccine and build a biomanufacturing center in Vancouver for the next pandemic. The company was bought a year in, bought by Danaher. Now they're part of the organization Cytiva, and I went full-time at Replicate, which I co-founded with a friend in 2020. So at Novartis, this was the vision for flu vaccines. So this is a state-of-the-art mammalian cell culture facility. It was finally commissioned in 2012, but they were building it. The operations were beginning. It's a massive facility, 475 square feet, 900 employees. You can see the fermentation trains here. These are 2,000-liter fermenters. We're at the top deck. When you look down, it's a couple of 100 feet down. And then you've got all the associated downstream purification systems. This is all clean in place. It's running non-stop. And from that facility, you can produce seasonal, pre-pandemic and pandemic vaccines. It was a co-development project with the US government. So if there's a pandemic in flu, it's designed to produce 150 million doses within 6 months of that declaration. And so we were then going to compete with that technology with RNA, was the plan. So this is state of the art in 2008, the in vitro transcription reaction. So for the first time, we have no cells. We do not need a facility like that built in Holly Springs. And the key questions to us were just, okay, what will the dose look like? How is it going to be 100 micrograms, 1 microgram or below? The lower it is, the smaller the footprint, and suddenly that facility can produce 28 different vaccines instead of just one. So you need a piece of DNA to run an in vitro transcription reaction. You need a promoter sequence for the T7 polymerase, and in comes the enzyme, and it will pull in a sequence-dependent manner the nucleotide trisephosphates, and off comes your RNA in a sequence-dependent manner, and it will just keep repeating and keep repeating. You could buy these in commercial kits, but nobody had really scaled them back in 2008. There was one little company in Germany called CureVac who were pioneering the space, but they weren't really publishing. So we began to explore scaling up this little this in vitro transcription reaction. And here's our, Geall from about 2009, showing the Ambien Megaskip kit we were buying off the shelf. This is a 9 kb RNA, and here's our at-scale kit-independent system, and the yields we're producing are around 5 milligrams per mil. This number, if this is not heard of in the protein world, so already we're getting excited. 5 mg per mil. This is the process. What it looks like when you buy one of those transcription reaction kits. Cap analogs back then weren't available, so we're enzymatically capping, and it's a batch process step by step. So you run your RNA in vitro transcription reaction, you then digest the DNA template, lithium chloride precipitate, wash with ethanol and resuspend and dry. You then come in, you denature the RNA, you cap it, more purification and done. This is not an industrial scalable process. And so we sat down and worked for about 8 months to figure out how do we get this looking like an industrial process. This is the original slide from probably week 1 of the project. Non-viral delivery of self-amplifying RNA. And this was our vision, reinvent the gene vaccine. Develop a synthetic vaccine with the properties of a viral vector without the complications of cell culture and anti-vector immunity you see with the viral systems, that was safe, scalable, and widely applicable, so we could drop in antigens from several different pathogens with the same process, have several different vaccines. We were lucky enough to have a viral replicon particle system in place. This is a packaging cell line that will take the same RNA we're using self-amplifying RNA and use a packaging cell line to put a capsid around it, an envelope, and the glycoproteins. It's based off alpha virus, and so this is viral delivery. We were trying to use a lipid nanoparticle system to deliver it and compare the two head-to-head. In the clinic, the alpha virus particle systems had been used. They worked well, they were safe, efficacious, and at about 10 to the 12 infectious units, great data. So we knew the RNA, self-amplifying RNA from alpha viruses replicated in humans from this system. There were just a number of questions. Could we even encapsulate RNA in a lipid nanoparticle given the process? Would it be stable? Would it launch? Were we using the right capping strategies? Would we get biological activity? So this is the alpha virus. It's a family of over 45 species now. This is the genome. At the 5' end, you have 4 non-structural proteins that make the viral replicase and allow amplification of the genome. In the middle here, you have a subgenomic promoter, and then at the 3' end, you have the capsid E1 and E2 glycoproteins. So when I talk to you about being able to make a multivalent vaccine, we already know that the authentic alpha virus is encoding this much. We've certainly synthetically been able to encode more cargo than this, and we're up to 5 antigens now in 2020. It's positive sense, so it replicates in the cytoplasm. So we only need a delivery system to get us to the cytoplasm with DNA, cytoplasm through the nuclear pore into the nucleus. So from a delivery perspective, it's a lot easier. And it's self-amplifying, self-replicating. Theoretically, we'll need a much lower dose. So it looks just like a regular RNA, shown up here, 5' cap, poly A tail, UTRs. The only difference is you've got these non-structural proteins, and then you drop your antigen in after the subgenomic promoter. That means with a single antigen, you're already out to 9,000 bases. If you put 5 antigens in, you're going to get close to 16,000 bases. We've worked very hard at Replicate. We now can do a GMP compliant production of a 16,000 base vector. And it's taken us many years of work to do that. We don't use base modifications at all. And so any of all the work I'm going to show you today doesn't contain a base modification. It's wild type RNA. But you know, through COVID and then after, the Nobel Committee awarded Drew Weissman and Katie Carrico a prize for their pivotal work in the field. We certainly explored it, but for vaccine applications, it didn't work. And what they're doing is moving from uridine to a pseudouridine or methyl pseudouridine analog. And that's what we saw going forward in the COVID vaccine trials. It allows you to dose higher doses. As I'll show you as we move forward, with these types of replicating vaccines, the doses are much lower. So now there are 5 different types of RNA. Back in 2008, we had one company, CureVac in Germany, with unmodified RNA. There's us with self-amplifying, it was called then, the SAM vaccine platform. Since then, base modified, trans-amplifying, circular RNA. I'm sure every year there'll be a different type of RNA appearing, but they all have the same principle, enzymatic transcription, figure out delivery. We can move forward very fast. So how does an alpha virus work? We'll start with linear RNA first. So here's your linear RNA. We get it into the cytoplasm. It engages the ribosome. It's encoding our antigen, and then it'll make a small amount of the antigen, usually over a period of 24 hours. Self-amplifying systems, once inside the cell, they replicate in what's known as a sphereole. It's a membrane-privileged bleb, and it's able to replicate for several days. the first thing it'll do is make a negative sense copy of its template, and off that it amplifies the genome, but it also launches the subgenome. This looks like endogenous RNA within the cell. It binds the ribosome and starts to make the structural proteins. The capsid will pick up the genome. The glycoproteins are on the envelope, and it will bud off live infectious virus. Because we've deleted the structural proteins, we can't do this, but we do replicate in the bleb. We make messenger RNA, but encoding our antigen, and I think all the examples are going to show you those are glycoproteins we put on the cell surface. So it's dead-end delivery, but it's mimicking a viral infection. So to the cell and cells that are transfected with the material, they are in danger signal mode. They're seeing that they're starting to, the RNA is amplifying, and the cell intracellular helicases at some point are going to detect it and say there's a viral infection going off somewhere. The lipid nanoparticle will traffic into the draining lymph node within hours and be picked up by the immune system. You get an innate immune response to that because it looks like a virus with RNA inside. And with that combination of events, it looks very much like a live attenuated vaccine. So it's very potent. We get strong antibody, strong T-cell responses. Half of my brain, I'm an engineer, and so this is how I think. These are the pillars of vaccine design in my world. And the antigen is the most overlooked content. So I always get excited when I'm in places where they're designing and discovering new antigens, because I can design new vectors, delivery system, and manufacturing. If we want new vaccines, we need new antigens. And so when I come in a place like this, I get very excited about possibilities of making new vaccines and getting them into the public and saving lives. So you need a very well-designed antigen. I'm going to talk to you about self-amplifying, self-replicating RNA, lipid nanoparticle delivery systems, and manufacture of those. So we'll start with RNA synthesis using the IVT reaction. So that transcription reaction kit, that schematic I showed, has got to change if we're going to go to commercial scale. Commercial scale for self-replicating RNA that I work on, we're less than 1 liter bioreactor, probably about 1/2 liter. So it's very small, very compact. Moderna, Pfizer, BioNTech, they're at about a 30-liter scale, so quite a large vessel. But here's your bioreactor. Everything has to happen in there. So you can't do stepwise production, intermediate purifications. So we had to quickly figure out how to do a one-pot RNA synthesis. So you make the RNA, you digest the DNAs, and then you come in and you cap it all in the one pot, no intermediates, just 1, 2, 3 steps, and then into purification. Certainly in 2008, there really wasn't much available. Now, through COVID, there's a lot of purification technologies available, built for purpose, ready for RNA. So you have to purify away from the fragmented DNA, the NTPs, the enzymes, any antibiotic that may have come through during E. coli production of the plasmid. But the purification levels that you need to achieve are monumentally smaller than anything from cell culture. We don't have any of the debris coming through from that host cell production line. So it's truly a lot easier right from the beginning. In 2008, we were looking at the residual content of the raw reaction mixture, and we were pretty close to an E. coli produced vaccine without any purification. So there wasn't much that needed to happen, but we had to get creative and figure out which contaminants were inhibiting immunogenicity. We need to dialyze and concentrate at an industrial scale. That's tangential flow filtration. I've been up in the labs and I've seen systems there, so you know all about it. And then sterilization. Yields, even at scale, were up to close to 8 milligrams per mil. So moving from the kit, we were able to scale quite well and keep production high. And we found that this is a generic process, small tweaks to the process when we change antigens, change vectors, but it's pretty generic. So LNP delivery, so there's a lot of, this is the first publication of an LNP delivered RNA vaccine back in 2012. This work was actually done in 2008, but we had to work hard with the patent lawyers to file patents. But we're encapsulating the RNA. Encapsulation efficiencies were very high, even with the first experiments. These long virally derived RNA sit, when we looked by cryo-EM and other techniques, were about 60 nanometers in a ball, phosphate backbone on the outside, bases on the middle, just ideal for encapsulating, a lot easier than linear RNA in most respects. In our crude first experiments, we were about 140 nanometers. Now we're down close to the size of a virus, which is at about 70, 80 nanometers. Composed of 4 components: phospholipid, cholesterol, they're the base stabilizing the lipid envelope, and a PEG lipid on the outside. We needed the PEG lipid in order to be able to manufacture the LNP. We've got to process it downstream through tangential flow filtration, sterile filtration. Without the PEG, it's too sticky, and we can't process it. The most important was this ionizable cationic lipid, and we'll come to it in the subsequent slides. I'll just say that for us, we had to publish a lot. A lot of the early team were postdocs. We had a few PhD students later on. So we pulled this ionizable cationic lipid from the literature from Peter Collis's group. It had been published. We had a big pot synthesized. And for everything we published, this is the lipid we used. But internally, we had stuff that was a lot more potent. But this worked pretty well, even from the beginning. So this is what the process looked like. This is our first prototype. It's a jerry-rigged syringe driver. We've got one syringe filled with the lipids in ethanol. The RNA is in buffer. We've got a dilution buffer, and we're shooting it through a T mixer. pH is low, pH 6, so that lipid is protonated. It's positively charged. The phosphate backbone's negatively charged, so the two lipids interact, and then the rest of the lipids pile in, and you form your liposome. We're at 33% ethanol. It's metastable, and you hold it for an hour. Everything stabilizes. The particles start to rearrange, and then finally you bring up the pH by diluting with a phosphate buffered saline, and then you're into tangential flow filtration to dialyze away the ethanol and change the buffer into your final formulation buffer. Now, because I'm at Novartis, we're all about scalability. We don't want to do spin columns or artificial dialysis bags. We were lucky enough that Spectrum Labs had just launched this new crossflow TFF system, and at a 2 ml volume, we could process this LNP. And so we could mimic a process at 2 mLs that we could do at 100 liters. And you can connect it up and measure transmembrane pressures and everything a chemical engineer wants to know about what happens as I scale. And so at a 2 ml scale, we could capture all the information and do predictions on scalability. We then filter it, 0.2 micron filter. From a pharmaceutics perspective, you need to be under 100 nanometers in order to be not blocking that filter. If you look at the distribution of your dynamic light scattering, if you're anything above 200 nanometers is blocking that filter. So you're running a volume through the filter versus time. Eventually the back pressures build and it will block. So this is what a process looks like now. You have a mixing device, and we'll come on to my favorite mixing device. It's just one example but it was a severe problem back in 2008 that needed to be solved. We are not moving forward with that syringe driver T-mixing setup. We felt tangential flow filtration, sterile filtration was saved, were safe. It's standard operating equipment in most labs at industry scale, but the membranes certainly weren't where they needed to be. Since COVID, we've seen prototype membranes being developed specifically for lipid nanoparticles and RNA, and that's a good thing for the field. So here we go, early proof of concept. It's all published in these 2 papers. There are several others. These are just my 2 favorite. The first one is we're tackling a respiratory syncytial virus. It's an envelope virion. The key target is this RSVF protein. Internally at Novartis at the time, there was a subunit protein, a vaccine being developed. So we had all of the protein design coming from them, that we knew it needed to be a trimer. They were doing fusion deletion approaches, and all of that information was available to us to drop in to our RNA vaccine. However, it's a competition. Whoever is the best goes forward into the clinic. And in order for us to have been chosen, we would have to be superior. Equivalence isn't going to count. There is no manufacturing infrastructure for RNA. Making a subunit adjuvanted with alum is Big Pharma's bread and butter. But this was the first target, not my choice, but it's a competitive environment in Big Pharma. So here's the first experiment. We're in BALC mice. We're vaccinating twice, 3 weeks apart, taking a bleed either post-first at day 14 or post-second at day 35. We're just measuring antibodies by ELISA specific to that F protein. We have naked RNA here. This is post-first. So naked RNA, LNP formulated, 10 micrograms down to 0.01 microgram. This is the control. So this is this viral replicon particle. So it's the same RNA, but we're delivering it virally. It's probably a picogram of RNA. It's pretty low. 1 to the 6 infectious units. This is at the upper level of the dose response. So this is a good control. If we can match it, we're in good shape. And we know that this works clinically in humans, the VRP. And so what you can see post-first, we were utterly amazed. All the mice seroconverted, and we saw a dose response. It's very flat because it's a self-amplifying system, but all the mice seroconverted. We then boosted them, and they all boosted. And at 1 to 0.1 microgram, we were equivalent to this VRP. And so we're very excited. Go read the paper. There's lots of T-cell responses. But for RSV, it's all about antibodies and neutralizing antibodies. So we went into head-to-head competition with the subunit. Now we're in the cotton rat model where you can do an intranasal challenge. Very similar setup, 2 doses, 3 weeks apart. We're going to look at some serology at day 35 and then look at viral titers in the lung after challenge. PBS as a control, naked RNA as another control, 1 microgram of RNA encapsulated in our LNP, the viral control system at 10 to the 6, and then the alum adjuvanted subunit at 10 micrograms. And what you can see here on the NUT titers, we're not at the levels of the subunit. We're equivalent to the VRP. Looking at antibodies by ELISA, again, we're below. But we're seeing good levels of protection. Really no difference between any of the vaccines. Based on this data and other data, there's no path forward for the first iterations of our RNA technology. That vaccine, the subunit vaccine, is the one that got commercialized by GSK recently. So it's very effective. But in order to win, you've got to be competitive. So in order to be competitive, we needed money, we needed to innovate. So I reached out through my networks at Novartis. I'd been told no so many times. There's no more money in the budget. Finally, the CEO told me, you can go get a grant. And so I went out and got a very big grant from the Defense Advanced Research Project Agency. It's valued they're $14.2 million, but it's a cost-sharing initiative. So as a senior research scientist at Novartis, suddenly I have $28.4 million to spend to innovate on the program. People were kind of sleeping during the writing of the project plans, not really paying much attention because I don't think they thought we were going to get it. And so what I wrote in there was we wanted to go after disease targets that there were commercial vaccines against, there were correlates of protection. And so we started to do studies in this contract on flu and rabies. And so, which was a little controversial because we didn't need a new flu vaccine because we had the new facility built in Holly Springs. But it was sold by me as mechanism of action, understanding of the technology. We'll understand it, we'll improve it, and then we'll find where we'll fit it. Oh, and so we were not alone in DARPA funding. CureVac, Sanofi, Incelart, Moderna, Novartis pumped over $1 billion into the field in and around 2012. And because of that money, the innovation happened, the data started to look much better, venture capital started spending, the banks started spending, big pharma got interested. So I can't sing enough praises to DARPA. They get no recognition for it, but we certainly have seen benefit for it. So the next target is flu. I think everyone knows here flu, the major antigen of neutralization is hemagglutinin. So we've switched from RSV-F now to hemagglutinin and dropping it into our self-amplifying RNA. Here's the first experiment. It's H1 from the A California swine flu strain from 2009. We are measuring hemagglutinin inhibition titers, so functional neutralizing antibodies. The target, again, the benchmarks are the material coming from that Holly Springs site, either with or without an adjuvant at the upper dose point in the mouse. We're dosing at 0.1 and 1 micrograms in an LMP. We're either doing a 3-week interval between vaccinations or an 8-week interval. The longer the interval between the vaccination, the stronger the boosting response with these self-amplifying systems. And this was one of the first experiments to do that. If you go to 16 weeks, the titers go even higher. So at 3 weeks, what we're seeing priming events happening. They're equivalent to the subunit without an adjuvant, but we're not at the levels with MF59. I think everyone's familiar with MF59 here. So it's a high bar in the flu world. We boost, our responses boost. All mice have seroconverted in that LMP formulation. As we've seen before, the data's clustering nicely. We're at the level of the subunit at 1 microgram, but here's MF59. We increase the interval of the boost, and now we're starting to differentiate for the unadjuvanted, but we're still not at the levels of the adjuvanted vaccines, but at least we're on target, and we began several years of optimization of the vector, of the delivery system, pushing the dose down and pushing potency up. None of that data has been published. But one of the true advantages of RNA was speed. This is the first example published in the literature of speed of the technology. So in 2013, in March, 3 people in China died of a novel bird flu, an H7N9. The Chinese CDC put the sequence online on a global sharing database. In the paper, you'll find 2 members of the Chinese CDC as co-authors, as a thank you for sharing data, allowing us to do global surveillance of flu. But we were collaborating with Craig Venter in San Diego. They just invented Gibson synthesis, so they were able to quickly synthesize the HA gene in San Diego, proofread it, check it, and then by day 3, it arrived in Boston into my facility. We cloned it into our plasmid. We grew up the plasmid. We made RNA. And so by day 8, we're showing here on an agarose gel, we're calling them SAM vaccines, self-amplifying message. The marketers by this point have got hold of us. We're not calling this self-amplifying RNA. We're not calling it a replicon. We need a name that's more friendly to the public. And so we're now the SAM vaccine platform. But here it is around 10 kb, containing that novel H7. There's a sequence online. There are no viral isolates in the US at all. We then throw it on cells, and we look on the cell surface of the cells and run a Western blot, and here's that H7. We had an antibody from an earlier, an H7N3 that cross-reacted, and so we've got full-length RNA. It's replicating in cell culture, and it's producing the antigen. We repurposed some mice from an experiment, and so by day 13, we vaccinated mice either at a 3-week interval or an 8-week interval. There are no controls available. There's no subunit vaccine existing. This is the first pandemic response documented. You can go read about it in this paper. We're measuring hemagglutinin inhibition at titers, naked RNA formulated 1 microgram. We have a negative control, an H1 HA I showed you previously. We shouldn't see seroconversion. So we're priming, we're seeing responses, we're boosting at week 3, 3-week interval, at day 35, we're above 40, the correlate protection. So the vaccine's working very well. If we boost at an 8-week interval, now we have titers up in the thousands. So now we're starting to be competitive. We have speed is an advantage for the technology. This is the last paper that I wrote while I was at Novartis. I wrote it with Jeff Ulmer, who I showed you that picture of. It was called Vaccines on Demand, Science Fiction or Future Reality. Here's a statement. It's basically saying RNA vaccines are the future for pandemic. And we started to argue about why that would be and production capacity. And so here is a graph. We're looking at human doses per liter. And as the dose comes down, if we can get to a human dose of 1 microgram, we'll be able to produce 5 million human doses per liter in a few hours. And so the site in Holly Springs are now getting very worried. I was thrown out of that building, giving a seminar, told I wasn't welcome, because they were going to have to eventually embrace a new technology and change is always very difficult. We calculated the viral delivery system, that viral replicon particle in humans, 10 to the 8 was the dose. It was about 0.5 nanograms. So from a scientific perspective, we thought 1 or 0.1 microgram, if you got everything right, was probably doable. We're never going to be as efficient during delivery as a virus with an LNP, but maybe if we optimize them, we can get within, 100,000-fold. These are all of the different species you went into at Novartis, the different disease targets. Much of it didn't get published. We worked with the US Army in 2003. Ebola had been weaponized by the US government, and they wanted a vaccine. And so we went into the aerosol challenge model. We cloned in the glycoprotein from Ebola. It was very effective, 100% protection. We didn't get to publish. I showed everyone the data on malaria from Yale yesterday. Just a list of all the publications. But I often get asked, well, why did it take so long to publish? Well, because in industry, you have to do a lot of work with the patent lawyers. So I got a patent attorney to do a search. Geall is the inventor. Over time, applications of patent applications, grants are granted by the patent office. And so what you can see, 2008, the program began. This is some of my work on DNA vaccines. You can see this huge spike here. And so I have a massive patent portfolio. I get no money. I signed off my rights. And it's nearly, you've reached this time of patent life, the cliff for these are 2028, 2030. But a lot of work from a lot of scientists to file intellectual property and a lot of experiments to enable these claims. As I said before, 2015, Novartis decided to get out the vaccine game. They wanted to focus on oncology, so they did some asset swaps with GSK. I chose not to stay. They were moving everyone from Boston down to Maryland. Moderna were hiring. Most of my team went to work for Moderna. I wanted to take some time out and maybe do something different and let the RNA technologies mature a little bit and do some consultancy work. And that's what I did. This is my list of what I thought needed to change when I left Novartis. So there needed to be better accessibility to lipid nanoparticles. A scalable mixing technology was missing. More potent vectors were needed. Reliable sources of GMP enzymes. Downstream equipment specifically designed for RNA and LNPs. CDMOs that could manufacture RNA to enable small companies to innovate. And there were a number of analytical techniques that needed to come along. COVID solved most of these. And so small companies like Replicate, we don't have internal manufacturing. We outsource it. And so we've gone from designing novel vectors, we began in 2020, to having phase 1 clinical data in this year. So it's very rapid now. The economics are much less. Building a clean room for RNA, $100 million. Now you can just outsource it to a CDMO and they can produce both the drug substance and the drug product. So this is the detour for 5 years while I was consulting. This is Replicate Biosciences. This is the first publication down here. It's a DAR1 antibody conjugate, highly potent siRNA. We're in multiple rare muscle diseases. I left as we were about to manufacture the phase 1 lot and file an IND for myotonic dystrophy. Now it's 2025. They're in registrational trials for 3 products. So it's been pretty rapid. The company is now public, worth over $6 billion. I was part of the early crew, the 25 people that invented the technology. We scaled it, transitioned it into the clinic. Very proud of that work, but it's a whole nother seminar. But during that time, I was chairman of the board at Precision Nanosystems. They became Cytiva. I helped them build out a lipid library that they wanted then to out-license to the world and create as research reagents, which to me was kind of democratizing the vaccine and gene therapy field. They also worked on a modern microfluidic mixer. This is the architecture they finally came up with, a toroidal mixer. You can change the diameter of these inlets and change the flow rates that can go through. So at a small bench scale, at 1 ml a minute, you can make your preclinical material, and then at GMP, at 200, 250 mils a minute, you can make 100 liters. It's the same particles, either system. So it's a seamless way of scaling. It took many years of work and iterations with them to get it right. This is the system we use to produce our clinical material. Here's just a cryo-EM showing these solid, dense particles. And whatever scale you're at, whichever of their devices, it produces the same material. Just allows us in a small biotech to really move quickly out from preclinical and get into the clinic and show the technology's working. So COVID came in 2020. I think everybody knows the story, but RNA vaccines are the only ones that don't require cell culture. I've shown you speed. It's fast. It's rapid. You can clone from sequences and move quickly. But we got incredibly lucky because we knew a lot about the antigen and we'd had previous coronavirus outbreaks. Here's the main front runners. CureVac had an unmodified RNA. BioNTech, Pfizer, and Moderna with their base modified. And then over here, some self-amplifying systems. All of these are in LMPs. These guys all fell behind because of manufacturing issues. Manufacturing longer RNA is very difficult compared to linear. The winners were in here with the base modified. We'd had outbreaks of SARS in 2002, MERS, this Middle Eastern variant, in about 10 years later. A lot of work had gone in from the vaccine research units, particularly at the NIH, on what's the key antigen. They identified the spike. They were making pre-fusion versions of it. And here's just an image, a cryo-EM image of that spike protein pre-fusion that came out from Barney Graham's group. So we got incredibly lucky, and the RNA vaccines won. If we didn't have that knowledge, we certainly would have had vaccines in time. And so antigen discovery and trying to survey what's out there is still very important. Just some timelines, pandemic is starting to happen and declared in December, and we're starting to have licensed vaccines less than a year later under emergency use authorization. Normally, vaccine approval takes 8 years. It's certainly going to slow down now. We're probably looking at 4 to 5 years. In the US, mRNA vaccines, maybe never again. We'll see. There's a lot of politics going on. This was the race in the self-amplifying systems, and we had a failure from Imperial College London. I'm just going to talk a little bit about Arcturus, a San Diego company. They probably changed their vaccine 4 times during the clinical process, optimizing the antigen, optimizing manufacturing, but they finally got there, and in 2004, they got approval of the first self-amplifying RNA vaccine in a lipid nanoparticle as a COVID booster. Here's the paper they published recently. They're comparing, it's a booster trial looking for non-inferiority. They're comparing against the BioNTech Pfizer vaccine at 30 micrograms. They're in at 5, so base modified versus unmodified. The safety profiles are pretty similar. They're seeing severe adverse events. This may be under emergency use. This is acceptable. In my world, we need to have a lot better safety profile. But they did see better durability, and so here we're looking at antibodies for a couple of different strains and looking at durability over time out to day 181. So showing superiority in the durability, and so that's now, a common thought within the field is the self-amplifying replicating systems will give you better durability and lower dose. The other thing that came from the pandemic, we started getting modeling systems. This one came from Imperial College London, and their modeling system is fairly crude, but here are the different RNA technologies. Here are the proposed doses, whether they're using modified bases or not, and the common size of a bioreactor you'd expect to use. If you have one factory, how long does it take to vaccinate the world? And no surprises, if you can get down to 1 or 0.1 microgram, it's going to be a lot easier. So, Replicate, we founded in 2020. I took a year sabbatical and worked with the group in Vancouver. I never went there because of COVID, but coordinated and got the lipid systems ready, the microfluidic technology ready there, and came on board in 2021. We're still 19 employees. We focused on developing these second generation vectors that are more potent than the first generation self-amplifying. I'm going to show you some phase 1 data. The sequencing data for the alpha virus species got dramatically better. It's one of the reasons we founded the company. We felt there was an opportunity to pull out novel vector designs that might be more potent, opportunity to file IP, raise money. It's the biotech mantra in the US. Most, if not all, those first generation use a common vector from Venezuelan equine encephalitis, TC83. It's derived from the live attenuated vaccine developed by the US Army in the 90s. They published a paper with the sequence before they filed the patent, and therefore that is freely available to everyone to use. But there are over 45 species of alpha virus. Sequencing data is better, so we start fishing out novel variants, getting them and launching them in cell culture, filing patents, and we found a way of optimizing the 3' and 5' ends to make them a little more potent and easier to manufacture. So here's some data. This is an independent study that came from Anna Blakeney's lab. She's a co-author on this paper. She had the TC83 vector. We're encoding luciferase. We sent her over an optimized V vector. So we're comparing species to species in the same LNP. They're dosed at the same dose. And we're measuring total flux bioluminescence over time. And what you can see is higher levels of bioluminescence and better durability. Typically, we see out to 30, 40 days for these self-replicating systems. When we went into a mouse model with flu, remember in the previous work at Novartis, we're dosing at 1 or 0.1 microgram. Here, we're dosing at 10 nanograms down to 1 picogram. We've either got an H5 variant from A Vietnam or the H1A California strain. We're doing a prime or a boost. We're measuring hemagglutinin inhibition titers. And so you can see the mice are priming even at these low doses, a little better for the H5 antigen. And then we boost for the H5. We're not seeing a dose response until we're down at 1 picogram. So we've pushed the technology. We've got a delivery system in-licensed from Precision Nanosystems. We've optimized the vector, and we've gone from 1 or 0.1 microgram now down to a picogram. We chose to use a CDMO network to not build the infrastructure ourselves. It was quite a collaboration. They weren't ready to go. So at Curia, we spent a couple of years teaching them how to make long RNA. And the way you do that is you have assays that tell you if it's working. We have a cell-based potency assay. We have a capillary gel electrophoresis system to measure integrity. And we probed the process all the way through to figure out where we were seeing degradation. Longer RNAs will hydrolyze faster, and time at temperature and pH are important. So we optimized the conditions to keep potency, and we did the same with Precision Nanosystems, who are now Cytiva, but we in-licensed the lipid from them so we didn't have to build that capacity ourselves. So we're saving time by in-licensing the lipid, outsourcing the manufacturing, but using our skills to teach them what to do. And the lipid systems we used went through safety tox testing, genotox testing under the Canadian government at the beginning of COVID. So this is the RBI 4000, our rabies vaccine. It's our V vector with the glycoprotein from rabies in, and we're comparing it to Rabivert, the whole inactivated virus, so the full protein repertoire. Why rabies? This was meant as a proof of concept for the platform. So in the US, nobody has rabies, so it's a clean patient population. Can we prime? Can we boost that response? In COVID, everyone is positive. It's a very complicated immunological interpretation of what's going on. So we chose not to do flu, we chose not to do COVID. There's also a World Health Organization established surrogate of protection in a new assay. If you're above 0.5 international units per mil, you're considered protective. There's a commercial product to benchmark against. So here's the trial design. 0.1, 1, and 10 micrograms, 2 doses, 8 weeks apart. Cohort 3 is the anomaly. It's a single dose. And then Rabivert is cohort 5. The standard CDC guidelines now are 2 doses for a prophylactic vaccine, 1 week apart. So we're dosing 8 weeks, 1 week for Rabivert. Primary endpoint is safety, reactogenicity, determining that dose for phase 2. Secondary endpoint is the new assay, and exploratory T cells. healthy people, 18 to 45, seronegative for viral infection, normal blood, no prior rabies vaccination or exposure. There were 3 people that were positive for rabies. They were pulled from the trial. I'll show you the data at the end. It's very interesting. It's published, so you can go read about it. But this is the overall summary of the data out to day 84. So in gray, this means in that group, they've got detectable neutralizing antibodies above baseline. Blue means they've gone to or above seroconversion for that surrogate of protection. So even at 0.1 microgram, we're priming the majority. We've got measurable responses. Some of them are protected. We boost at 0.1. The majority are protected. Nearly 100% have seroconverted. If this was COVID, this would probably be viable. 0.1 microgram, we're at 50 million human doses per liter in a few hours. So this changes the world of pandemic response. 1 microgram, 100%, 2 doses. Nearly all of them are protected. Even a single dose, conventional linear RNA with base modifications, if you look, they're barely priming in that NEUT response. Here, with a single dose, we've seroconverted everybody. They've got NEUTs, but nearly all of them are protected. 2 doses, 100% protection, and we can match Rabivert. So here are the kinetics out to day 84, and you can see no Rabivert comes up, prime and a boost, and then the titers start to decay. Our RNA titers come up, and then we boost, and they all come up nicely. We're monitoring out, we're just rounding off the data sets to day 240. It looks like 2 doses at 10 micrograms are superior in durability to Rabivert. If we look at post-prime, what happens with the RNA at day 28, where we see the maximum titers, we see a nice dose response in the NEUT assay. Here's the correlate of protection. There's that one that didn't seroconvert. We'll kind of come on to the safety data, but what we see from the safety data is no dose-limiting tox, no severe adverse events, so we could dose higher. So in phase 2, we're going to continue this dose escalation. Can we dose 15 or 20 micrograms? Is it tolerated? And can we push a single dose to the level of Rabivert over here? So when we're boosting, this is Rabivert at day 18, and here's our 2 doses of 10 microgram at day 56, the maximum titers we're seeing. Statistically, these are equivalent. Obviously, these guys here seroconvert at a different time point. So we're starting to look very competitive with a commercial vaccine. We've got the opportunity to do single dosing. We're in a place where other RNA technologies are not. Safety-wise, this is the local reactions, either first dose or second dose. The thing to note is there are no grade 3 reactions. There are certainly no severe reactions or any dose-limiting tox. It's a small patient participant group. It's 18 people. If maybe the second dose looks less reactive than the first. This is certainly not what we see with other platforms. Usually reactogenicity goes up. But they're very mild, transient, and people recover within a few days. This is systemic reactions. We saw one elevated temperature in the middle dose cohort. It was reported from home, 103 temperature for 4 hours. There were no other symptoms. The participant got the booster, reported no adverse events at all. The physician thinks they misread the thermometer. It's reported. We don't think it's real. But overall, grade 1 and grade 2, and again, those drop on the second dose. So from a vaccine perspective in the mRNA world, we've got potency, we've got great tolerability data, and we're very competitive. 3 people were positive at baseline, which was unexpected for us. They got the first shot, but what you can see is we got a dramatic increase in titer, 13-fold. There was 1 in each dose cohort, and the titers remain very high, haven't come down, and we continue to monitor them. Here's the surrogate level of protection. No. We think. So I know you guys have been exploring prime boost strategies. And certainly it's a way of dosing, vaccines. You prime with one antigen, boost with another, or you can mix them together. But we were surprised by the data. We don't know why they were seropositive. Our assumption is they were in the military or a BH-TEC assistant and they've been given a conventional vaccine. But we love the data. No dose response, 0.1, 10 micrograms, pretty similar, and they keep going out over time. So overall, clean safety profile. We're in very good shape to continue the program. We're not continuing the program. We're in active discussions with people in South America, potentially somebody in Brazil, and hopefully we can get the program through phase 2, 3 and licensure, both as a prophylactic and a post-exposure strategy for South America. But we're definitely seeing improved bioactivity, potential of a single dose, everything that we could have hoped for improvements in the technology. So overall, how do we compare to the other vaccine? Most of the data's in COVID. We're in rabies. So the easiest way to compare is therapeutic index, highest clinical dose reported with favorable safety profile, divided by the lowest clinical dose showing 50% protection, and greater than 100 because we didn't see any tox, and everyone else, whether they're self-amplifying with modifications, without, they're all around 3x. So they're all dosing as high as they can till they hit tolerability issues, and that's their dose. That's not where we're at. We think we're in a competitive space. This is our pipeline. So we're hoping to move rabies forward. Our lead program is in Epstein-Barr virus. We're going to go in and treat infectious mononucleosis in the adolescent population. This has 5 antigens, so this is pushing out to 16 kb. This is where we think there's a unique opportunity with the technology. 1 vector, 5 antigens or 6 antigens. And hopefully we can optimize the cassette and the sequence and how we express the antigens to get equal potency from all of the antigens. We have some undisclosed targets. We have some preliminary data in inflammatory disease. We have a breast cancer vaccine parked up, ready, waiting for funding, where we're going after mutations, known mutations that occur in estrogen-positive breast cancer when they're on hormonal therapies. And so if the cells mutate, T cells will take them out. With that, just like to say thank you to the 100,000 probably people I've worked with. This is the team at Replicate. Thanks to all the study volunteers, the site personnel. Anna Blakeney Acredo are up at UBC. Hopefully I can get them talking to you. She's a self-amplifying RNA expert, knows about delivery. She'd be a great place to send interns to do training on. Thanks to the clinical people, clinical sites, and our CDMOs who made the material. And that's me, that's my wife. Somebody's taking a picture of me taking a picture of the turtle. Yeah.
Okay, thank you, Andy. Do we have questions?
We've been discussing for 2 days now, but I still have few questions regarding the optimization of the vector.
Yes.
Because that's a big issue. If you want to have like 4, 5, 6 antigens, do you really need all 4 non-structural proteins? Because only one is the RNA replicates, isn't it? Why you need all 4 NS proteins?
The biology is quite complicated.
I'm sure
you've got to
guys are
cap, you've got to put a poly A tail on, you've got to get it to cleave off the subgenomic promoter. But those proteins also go into the host cell genome.
Yeah.
And they co-opt, and you suppress antiviral responses, you skew protein expression towards the viral genome needs. So they do a lot. But we now have 17 sets of non-structural proteins that do different things. So if we want a T-cell vaccine, we screen, and it's usually not V, it's something else. And
yeah, I was wondering if you can make some sort of deletions in this NS protein to make the whole thing smaller, and it not
sometimes
affect.
one deletion kills,
just kills
the whole thing. And so people have been trying for years to do that, and we've just said,
that's the way it is.
we're going to go with a library of different vectors with different properties and explore that rather than trying to do point mutations or...
Okay. And second quick question, how much plasmid DNA you need to start your bioreactor? Like for 1 liter, how much DNA you need to put on?
It's like a 1 to 300 conversion. Yeah, it's something like that. I don't have that number. No, it's not a huge amount. Yeah, in a liter we can make 5 grams of RNA. So it's about, you divide by 200 or 300, and that's the amount of plasmid you need. But we're still, we're always optimizing the plasmids, and so now we're looking at synthetic plasmids where they're a lot simpler, that you haven't got any of the spinach you need to grow them in E. coli. Maybe we'll need less of them. They're not quite ready for 916 kb yet, but it's coming, and then it's a lot faster. For us, it's 3-4 months to make a GMP compliant batch of plasmid. Synthetically, you could do it in probably 3 weeks.
Thank you very much for the presentation, Dr. Geall. I'm a pharmacist, not an immunologist, so
I'm a pharmacist.
I'm going to ask you something about the RNA production. How much more expensive or more complicated is to make this self-replicating RNA compared to the mRNA?
It's the same recipe.
Okay.
You just got to play around with time and temperature and the sequence of addition, and it's very similar. And we're kind of I'm back and forth now with some commercial enzyme producers, and they think if a dose with the let's say it was 10 micrograms, they think they can get below $1 for. So we think the cost of goods is going to be very low because the doses are lower, and our aim is to try and get to less than $1. So that then it's cost effective in South America and beyond, because we can't be charging $100 for a vaccine here.
Yes.
$10 would kill everyone probably. It needs to be a couple of dollars.
Okay, and the other quick question is, in some point in your presentation, you showed when you were doing the consulting work, you listed the challenges for developing the vaccines by that time. But now, what would you consider the biggest challenge that we have to face to expand the pipelines of mRNA vaccines or RNA vaccines?
Antigens. Good antigens.
Antigens?
Yeah. Okay. So antigen discovery. So CMV is a huge unmet need in the developing world. I think it's 96 proteins in the genome. Which one? Which one do you need for the perfect vaccine? So it's always antigen design and finding the right antigen to go. So I think we have a great platform, and it's now for us, it's finding the right antigens to pair it. We're not going to be able to tackle every infectious disease, but there are many we can tackle quite easily. For instance, seasonal flu, putting in 4 HAs, not a problem. But if those HA constructs are not designed well, the vaccine won't work.
Thank you for your presentation. It was really good. And I have two questions. The first one is, for how long does this self-replicating mRNA they still there in the cell? And could you measure the amount over the time?
So in the mouse with luciferase, we see bioluminescence for 30, 40 days. And the half-life of luciferase is hours. So, but when we put an antigen in, it's much shorter. So there's a published experiment in 2017 from the team at Novartis where we did luciferase alone, we did luciferase with a bicistronic with RSVF, and the bioluminescence truncated down. So when you put a glycoprotein on the surface, the T cells are going to see it. They're going to come in T cell positive for the antigen around day 7, and that's when you start to see clearance of those transfected cells. In cell culture, 3 or 4 days, and they go into apoptosis because it spills out into the cytoplasm, and it goes into apoptosis.
Yeah. So the second question, it was depending of this answer. So do you think that you can extend the duration of this
yeah, that's
mRNA?
one of the reasons we went in to look at the different species. They all have different patterns of the way they replicate, and some of them replicate at a lower level, but they go on for months.
Doing that, do you think that you can use this kind of technology to deliver proteins to cells?
That's so behind the vaccine engine, there's a gene therapy engine where we're trying to come up with ways of expressing antibodies. So the cell line development for a commercial antibody is years. From sequence in the RNA in your arm, make it endogenously. It would be much faster. We could, and so, if there's a future pandemic, we're thinking in the hot zone, you're vaccinating with the antibody system, and then around the outside, you're coming in with a vaccine to induce immunity.
And as a therapy to challenge those genetic therapies that we have now, do you think that's feasible? For example, we have CRISPR-based genetic therapy that will correct some.
Yeah,
So if
CRISPR is a linear RNA specialty. They need to be in, express, done. So it's short and sharp, and you get your effect. This kind of self-replicating systems, they're around for too long, and the gene editing systems, linear RNA with the guide, it's all about delivery. They need to move away from the liver now and find new targets, and the delivery is the challenge, and it's going to be a tough challenge.
Yeah, good. Thank you.
I think they ask my questions most. But going back to the antibody therapy, so do you have to have some type of a tag that drives the RNA to B cells, or you think that any cells you put that RNA would
the early experiments are just IM injection, and the muscle cells are able to produce
lots of antibodies.
Yeah, and you code in the heavy light chain. The liver does a much better job. So a lot of people have gone systemically and got production from the liver. They're better designed to produce antibodies.
And you can measure high levels of antibody in the
They're not high enough yet. So with some of the really potent antibodies, serum concentrations can get up to levels of efficacy. But it's going to be 10 years of development. We can probably get to the levels, but we've got to avoid immune stimulation and generating antibodies against the antibodies.
The other question I had was the cost. You already answered. So do you see what would be the main challenge in terms of regulatory issues to approve the self-replicating
Yeah, it's going to
RNA
be
as a product?
It's going to be perceived risks. So, you know, how long does the RNA persist for, which we can measure. What happens if is usually what happens if you get a co-infection with an alpha virus? Could they combine? And so, and there are lots of studies showing that doesn't happen. You can induce it in a cell culture system where you massively overload the system, but the alpha viruses super infection exclusion happens, so one tends to replicate in the cell and not two. So, but we have to address them and we have to generate data, and certainly for phase 1, everything went well, but it's an ongoing dialogue with the regulators to understand where, and the public are going to react completely differently when they hear the word self-replicating, and so we've got to educate them that this is a live attenuated viral vaccine. This is a lot safer, and why, and convince them and generate safety data and keep moving forward. And it'll take time.
We have a question from the internet, Daniel Doro. He's asking if there is any limit to how much info in base pairs these vaccine constructs can carry. You said that.
So yeah, there's no limit in terms of the plasmid that we've seen yet, but in terms of manufacturing, a GMP compliant process where we have to have defined integrity of full length and maintain potency, 16 kb is what we can do. So that's about 9 kb of coding sequence, which is a lot. There's a lot. Yeah, there's a lot we can do with that.
Way more than what we are used.
Andrew, I was discussing with Sarah, if we use non-modified mRNA, it's important to purify, to remove the double strand, right? Because otherwise the immune response will be too strong, right?
Yeah.
Okay.
Well, so when you dose low, it's a dose-dependent effect. So, and for vaccines, it doesn't have to be 0. And so what we've found is there's 3 ways of achieving low double-stranded RNA. One of them is you purify it out. I think one of the students was showing
the cellulose method.
Cellulose, yeah. Some of the modern enzymes, T7s, have much lower. The yields are much lower though in the IVT reaction, but I'm sure that'll change. The other one is if you can measure it, you can optimize the IVT reaction, so you can change temperatures and times and concentrations of things, and the level of residual double-stranded RNA will go up and down. And so you can reduce it 3 ways. For me, at the doses we are, it's easiest just to optimize the IVT reaction, get it down to a level that is already acceptable.
Okay, thanks.
And we're lucky we have data where we know it's acceptable. And so with our assay, and the problem is everyone has a different assay, there's no standards yet. But we now, as we switch programs, we now know what residual levels are okay, and we'll just maintain those levels going forward.
Okay, thanks.
Okay, I think we are good. Andrew, thank you very much again for your generosity, donating your time to be here. 3 days, 5 lectures. We learned a lot, and we hope that this will be just the beginning of long-lasting collaboration.
I do, and we're going to work together and conspire and figure out how to make things happen. I was overwhelmed by what you do here. It's very impressive. It's like end to end. You do everything, and with the new facility, when when you're capable of GMP material going to phase 1, it's going to be amazing.
Yeah. Thank you so much.